A very warm welcome to the audience to this session of Nobel Fest entitled Rooted in Genes, What Can We Learn from DNA? My name is Gonzalo Hortelano. I am a professor in the Department of Biology at Nazarbayev University in Astana, Kazakhstan, and I am the moderator of this session for you. Now, during this session, you will be given the opportunity to ask questions to our speakers. So please use this and send us our questions so I can put them through our speakers. Today, I feel extremely fortunate to have the opportunity to join two speakers. They're two exceptional geneticists, two truly giants in the field. Let me first introduce them to you. Hello, Professor Lap-Chee Tsui. How are you?
Good, I'm fine.
Joining us from Hong Kong. So, Professor, the Honorable Lap-Chee Tsui is a world-renowned molecular biologist. He is the current president of the Hong Kong Academy of Science, has been the Vice Chancellor of the University of Hong Kong, and before that was geneticist-in-chief at the Hospital for Sick Children in Toronto, Canada, and also a university professor, the highest rank at the University of Toronto. He has received multiple awards. He's a member of the Academy of Science of in the US, London, Canada, China, and he has received numerous awards, including the French Légion d'honneur. But his perhaps the best known contribution of Professor Tsui to genetics came in 1989 when he identified the gene that causes cystic fibrosis. This was a breakthrough in human genetics. And in addition to that, so that paved the way to start the Human Genome Project as well. So he contributed also to other diseases and has been a key figure in genetics ever since. He probably doesn't remember, but I met him at Hospital for Sick Kids in Toronto shortly after he cloned the cystic fibrosis gene. His clear vision of science is only matched by his also very well-known humility. Professor Tsui, welcome to Nobel Fest.
Oh, thank you.
We also are fortunate to have Professor George Church joining us from Boston. He is a professor at Harvard, but also leads synthetic biology at the Wyss Institute associated to Harvard. This is an institute for biologically inspired engineering that uses biological principles to develop new engineering innovations. He oversees the directed evolution of molecules, polymers, and whole genomes to create new tools to study genetics, to sequence genomes faster and more accurately. He has contributed numerous ways. He has been pivotal to start and complete the Human Genome Project. He has contributed the concept of molecular multiplexing and many other concepts in genetics. He is a professor at the University of Massachusetts Institute of Technology and Harvard as well. And again, he has received multiple awards for his many contributions of science. And he is a member of the National Academy of Science and Engineering. Professor Church, you have contributed in so many ways to the development of genetics. Welcome to Nobel Fest.
Thank you very much.
When you started working on the Human Genome Project, Dr. Church, did you imagine how far and how fast the field of genetics would actually take us?
In a way, yes. I mean, in that I had hoped that the Genome Project would be a technology project, and it was fairly clear that technologies could improve exponentially. I don't think it was very specific. It ended up not being that much technology until 2004. But in 2004, next generation sequencing developed, and that was even more surprisingly fast.
Yeah, yeah. We'll perhaps come back to that later. I was curious to ask Dr. Tsui, when during the cloning of the cystic fibrosis gene, and once you realized that the protein is involved in the exchange of ions in cells, and therefore clearly explaining the symptoms of patients with cystic fibrosis. Was there in that story some kind of eureka moment that you see, this is it?
No, this is really, everybody thought, yes, I found the gene, I know exactly what it does. No, we found the gene by genetic means. That was really before the Human Genome Project. When we had the gene, we did not have the whole gene. We pieced together the gene bit by bit. And when we said we had a gene, we had only one single mutation at that time. Not to go back that far, but then of course later on we knew there were many other mutations, and confirming that indeed this mutation in this gene causes fibrosis. But at the very beginning, we had only a mutation telling us, yes, we got the right thing, but you talk about this small chloride channel in million of secretory cells. That was really a lot of work behind it. Before we cloned the gene, there was already a lot of people speculating that this could be a chloride channel defect, because a lot of people already did the physiology and did the measurement and compare healthy and diseased patients. And so that was a hint. But then to link the gene to that particular observation and saying that the gene product is a chloride channel took really a long time. A lot of people put effort in there. Therefore, there's really no eureka moment to say, yes, we had it, it's a chloride channel. No, there's a lot of work behind it. In fact, we had nightmares, if I can digress a little bit. A lot of people claim a lot of, let's say, discoveries, and later on, they did not have enough evidence. So, although we submitted the papers to the journal, and then it was accepted, or the papers were accepted for publication, there's a good chance that the conclusion we had was wrong. Okay, so we had nightmares, and some people might not, but I did a lot of nightmares of what happened if we were wrong. So no eureka moment. So, sorry, a long answer to your very simple question.
Yeah. So actually, you in both cases, certainly in the case of cystic fibrosis, there was an enormous contribution from a lot of people who really sort of made that possible. Chromosome 7 is a large chromosome, so it was a big undertaking. And the same with Dr. Church, where basically long gone the days of single individual achieving a big feat, and now it's more large teams that often at the multidisciplinary fields that contribute. Dr. Church, you want to comment on this change in science, not just genetics?
I'm not, I mean, I love teamwork. It's fun working, even if the team is just two people. I'm not convinced that large teams are inevitable. I think that the next generation sequencing, for example, an individual person can operate a simple machine that does DNA sequencing. In fact, some of them will fit in the palm of your hand. So certainly my efforts are to try to bring down the costs of just about everything, DNA sequencing and synthesis and synthetic biologies, to the point that individuals can make achievements. Even do-it-yourself biology is possible. So like I say, I love teams. I love even big teams, but I also work hard on making technology accessible to individuals.
Yeah. I mean, I think you agreed that the developments in genetics since Watson and Crick published the DNA model back in 1953 has been nothing short of spectacular. And basically at the level we have achieved today is something that not long time ago would be almost like science fiction. And not only with applications or implications in biology and medicine, but also in other very diverse fields like justice or even insurance. And not only in theoretical knowledge, but really in applications. And I think Professor Church will sort of talk a bit more about that in a minute. But just as give the audience a sort of a small example of what I mean by this, I'll give you an example that's really close to me. So I have a son who is a professional athlete. He's an Olympic sprinter in athletics. So normally athletes train to exhaustion and then they rest and that's it. But my son had the genome sequenced, and particularly looking at genes involving his physiology, energy system, muscle, biology, nutrition. And so what we learned from that can be used to personalize his training to better fit his physiology, to basically also adapt his diet to take into consideration the nutrients that he assimilates, or he really cannot assimilate that well. So this is one single application today. Sorry, yes.
I just want to bring a cautionary note. Okay, just like a disease, we thought we know many mutations, and mutation causes cystic fibrosis, mutations causes and all that. Okay, having a mutation is not enough to develop a disease. In fact, a lot of people are out there with mutations without symptoms. Okay, so there are other genes there that would compensate or actually change the properties of the original properties of your gene you're looking at. So genes work together. It's not just one gene, one activity, and 5 genes, 5 activities. Okay, one gene, maybe a set of activities, 5 genes. You know math, right? Is the order much higher? So it's not just simply that you identify these gene sequences, then you can be an athlete. Your son, in this case, he has certain genes, okay? But there may be a lot of other people have the same genes out there, but they're not athletes. You cannot say that, well, you have the genes, you'll be athletes. Even though with training and so on, it's not the same. That's one point I want to make. Sorry to interrupt you.
Yeah. The sequencing of the human genome was a milestone, in many ways without precedent in biology. However, perhaps obtaining the 3 billion base pairs of a genome and collecting a sort of large amount of books in it was not in itself the main goal. And so comparing, let's point out, a variety of genomes allowed us to learn more about the structure and genome function. The next goal, however, is perhaps the personalized medicine, how to use that to be able to treat a patient based on his or her unique genome. Professor Church, how far do you think we are from basically achieving such an ambitious goal for general population, not just specific people?
Well, I think we're already at a point where many people could benefit. Possibly $1 trillion could be saved by carrier screening. and lots of not just economics, but terrible psychosocial burden on the families these are very severe diseases where modifiers are relatively rare, and even if and the cost of false positives are low when you're doing carrier screening and preconception genetic counseling. If you wait until a person is an adult and you want to treat them, then false positives becomes a much more severe problem. For example, if you have a cancer predisposition and you get radical surgery, say mastectomy and ovarectomy, and that was a false positive, that's a medical tragedy. But if you're getting advised to avoid a certain carrier status, that has them lower. But anyway, the point is that is already in practice, and could easily impact. It could be practiced on every single person considering marriage or childbirth. There's also non-invasive prenatal testing, which is also preventative and personalized precision medicine. So I think those two examples are the big ones. And then there's some for cancer where one might use engineered cells like CAR T cells or something like that, where you're custom making a particular category of T cell to attack a particular category of cancer cell.
Perhaps we are looking at the genome as something more perhaps dynamic than we used to, like over the lifespan of an individual, in terms of contribution of epigenetics and so on. Any your comments on that?
Yes, I mean, certainly developmental biology and aging are part of the same process and are getting to be well understood in terms of epigenetics. We now have a complete set of transcription factors our lab has developed that allow us to go from essentially any point in development to any other point in development, at least at the cellular, sometimes at the tissue and organ level. That doesn't mean we understand it well enough to, I mean, it's getting into, that sort of information is getting into the clinic. But there's a great deal, it's sort of a half empty, half full cup. There's quite a bit we know, quite a bit we can do without full understanding, but full understanding is quite a ways off. But we can do engineering with considerably less than full understanding.
Yeah. I remember the classic view of genetics that DNA codes for proteins, and as an undergraduate student, then you could see the coding DNA and then junk DNA, so-called junk DNA, that apparently serves no purpose whatsoever. Now, the Human Genome Project revealed that only a small proportion of the DNA actually codes for proteins. It's a very small proportion. And so the vast majority is supposed to be junk. Now, of course, Professor Tsui, can you sort of give us your view on the evolving view of the genome from coding but non-coding and the so-called junk DNA?
Well, if I can tell a joke, actually, it's not useful. You throw it away. It's called garbage. And for things you keep them dearly, they're called junk. Okay. Therefore, junk has some meaning. Okay. Just you don't know what they mean yet. In fact, the whole genome project and most sequence are compiled. And then when talking about the transcriptomes and looking at the RNA. And now the central dogma from DNA to RNA to protein more or less has been broken. Okay, you have reverse transcription from RNA to DNA, and we haven't got protein to DNA yet. That maybe you should tell us something about that. But still, I mean, RNA is not, the protein is not the only workers inside a cell to perform function. RNA can perform function. We know that like ribosomal RNA has been all been there, and then it play a transfer RNA, doing a lot of things at the RNA level. But now the so-called junk sequence, in fact, there are a lot of these non-coding RNA and a lot of microRNA that have been categorized. But sometimes I say the information has been accumulating. I don't even know. There may be more RNA species or RNA genes, I'll put it that way, than protein out there. But then the function of these RNA genes are not entirely clear. It could be protein genes in evolution. It could be things that by mistake they made a lot of these junk RNA, but still some been shown to have real activities. So therefore, I think the information is accumulating. It's just that protein coding genes, it has a product that we've been studying, or scientists have been studying proteins. So before we actually know much about genes, okay, or RNA, and therefore thought DNA, RNA, and protein. Now the understanding of biology is entirely different. But I must also caution that that there's so much information out there that we really need a lot of work to understand more. The human genome is out there, but I dare say that no one's genome is sequenced in full and aligned in full. There's still a lot of gaps. The human genome is out there with long sequencing. George must be working on that. But still, I mean, a lot of sequence you can see out there is assembly of small pieces placed together. They're not really genome, it's just sequence information. And then sometimes you don't know where that sequence is, and then there's a duplication, there's a lot of the arrangement of the genome is not easily understood. So therefore, when we talk about the human genome, the sequence and so on, there's still a lot of work to be done.
Sorry, I couldn't quite hear you. Could
Sorry.
you please repeat what you said?
My husband is doing silly things. Sorry about that.
I'm sorry.
Sorry, let me turn it off. Yes, please. Yeah, George, maybe you should take that.
Oh yeah, so I think we will have a complete genome or a method of producing complete genomes telomere to telomere without any gaps very soon. We're already seeing the first 1 or 2 genome chromosomes done out of the 23 chromosome pairs. So I think we already have ways of haplotyping over almost full chromosomes, but there's still hundreds of small gaps. And that will be inexpensive. We brought the price down already 10 million fold. It'll probably be 60 million fold by the end of the year. As the quality improves, you still need additional efforts to determine the functional significance, but we don't need to be able to interpret every part of the genome. Although it's very reassuring to be able to read every part of the genome, even if we cannot interpret it, because there could be things hiding, things that could be quite significant. So we want to at least know what people have, even if we can't interpret every piece of it. The junk DNA, I wrote my PhD thesis on the junk DNA in introns, so-called junk, and it turned out that one was an immunoglobulin enhancer and the other was a splicing enzyme. And this continues. So I think there's a lot of excitement on what used to be called junk.
Professor Church, you have worked tremendously on synthetic genomes and based on the understanding of our genome and others. This clearly would be key in our better understanding, of course, of our own genome, but could you also use this information, or could someone use this information to basically optimize human genome in and this is a question from the audience, can we use that knowledge to basically alter genomes in a way to make either, perhaps more sort of physical attributes or physical appearance, maybe intellectual? So your comments on that.
Well, I think many things are possible that are not, that never quite make it to the market, never make it to adoption, or there can be possible without being popular. So I think it's also, when you say optimize, you use optimizing towards a certain goal. There is no perfect means of transportation. Bicycles and super tankers, they have different optima. So you can only have local optima generally for an engineering goal. People do what you would call elective medicine, elective surgeries that are not essential. That will probably be true for genetics as well, as it becomes safer, more routine, more predictable. But I think that the elective surgeries are kind of, I think, a fairly small category, and I suspect that may also be true for genetics. But we need to certainly have in place very strict ethics, surveillance, watchdog, whistleblower, all sorts of mechanisms for making sure that this goes just like other pharmaceuticals or better.
Yeah. I remember Dr. Tsui, shortly after the gene for CF was cloned, that people immediately thought, well, all you have to do is inhale it and that's it. We'll fix cystic fibrosis in a couple of years. You probably remember that. So 30 years later, we're still struggling to get to that. Of course, there has been some advances, particularly lately in cancer. In fact, the COVID-19 vaccines currently in the later stages of development are actually gene therapy vaccines. But nonetheless, will we be able to one day conquer diseases and have this sort of sought after disease-free utopia?
Well, we may actually get in here because you mentioned about the cloning of the cystic fibrosis gene. Immediately people thought about gene therapy. Okay, you know how many cells we have in a body? 38 trillion cells. And then the cells all perform very different function. Okay, even though in the same piece of tissue, there are different cell types in there. So you put a gene in there, if it's not regulated, the gene may perform an undefined function, and then you don't want that. Okay, so, but knowing the disease gene, okay, we're talking about genetic diseases here. Cystic fibrosis is a simple genetic disease, autosomal recessive. So you have a change in a gene or a mutation in a gene. You have two copies of it. You have cystic fibrosis, and there are many other diseases of this type. Simple genetic diseases. Knowing defect, knowing the gene defect, or knowing the gene that causes disease, will give you really a clue where to look for the problem, how to explain the disease, and find new treatment regime. Okay, you cannot. Well, of course, now people talk about gene editing. I can come back to that later. But finding the basic defect of disease just give you a way to treat patients better. Okay, at least that's a thing that we talk about all the time. We want to treat the patient, therefore we identify the gene. Okay, so you know what? 30 years later, it's not gene therapy that cures cystic fibrosis. We can talk about gene editing. Gene editing hasn't come yet. Okay, and in fact, gene editing, the technology is possible. It's all out there. It's very easy. You buy a toolkit, you can do gene editing yourself. Okay, but it's not recommended. Okay, at this time. Then coming back to cystic fibrosis, Scientists has been, not myself, okay? I retired from science 20 years ago. So, that's like that. But anyway, so scientists have been studying this chloride channel, finding ways to correct the basic defect. And you know what? More molecules people have not seen before, can now come in to interact with this protein product, the defective protein product, to make it active again, make it fold properly, make the channel work again. So patients are treated by small molecules that have not existed before, and then it's highly effective. Patients treated with these small molecules, they behave like normal individuals. How wonderful. So I'm really sort of want to make a point that knowing basic defect is the way to treat the patient. There are many ways to do that. Now come to gene manipulation. Now, people talk about gene splicing or gene editing. Okay, so somatic cells, you have to change the cells, to change the DNA in the cell very specifically. Now the technology allows you to do that, but there's simply too many cells in the body, too many cells in the tissue. Okay, if you put the gene in there, okay, you can of course change all the genes in the cell, but the cell is going to die, and the new cells will come up without the gene. So now people talk about treating the stem cells. Great, go a little bit further, bring stem cells. But stem cells may not just make a cell for that particular purpose. They can be many other cells. Okay, of course, gene editing, even in somatic cells, can cause undesirable effects. It may cause cancer. Of course, you can say technology can screen them away, okay, before you find the cells back to the patients. So things like these are happening for certain diseases that are not treatable, there are no drugs and so on. People are doing experiments. But like George was saying, that we have to be very cautious when you apply this new technology to human health. There are lots of ethical considerations. Coming back to change the gene so that you can be a better individual. Wow, that is a very serious matter. Okay? So in theory, it's doable. I mean, the genome editing forum conference happened in Hong Kong some 2 years ago, more than 2 years ago. Okay? And someone, okay, announced that he made some babies with genes corrected or genes changed. That caused a really uproar in the scientific community. And therefore now there are the organizations coming together saying that one should not really do that until the technology is much better understood. And then to apply technology, one has to look at many issues, and the ethical issue, of course, is number one. And then Gonzalo, you started the forum by saying that genome research, DNA research is not technology anymore. I think George said it. It's for humanities. Okay, you talk about really how we look at the life of a human being, and then how we treat ourselves as a race.
Yeah, I think I agree with you. The danger of underestimating the complexity of biology, and just to be very cautious that, and perhaps also to learn from the past experience moving forward as well. I would like to put forward a question that I got from the audience, and it's a question to Professor Tsui. Do you think we will be able to see a living superhuman in our lifetime?
A lot of superhuman beings around.
Whatever that is.
Genius, and then there's by natural sort of ability, and then with I must come back to nature and nurture. Okay, there are many geniuses around, and some math genius. They did not go to school. They were able to scribble the mathematical formulas on a tree bark all by themselves. Okay, So these are truly genius. Okay, the genius comes and go, and then very difficult to create genius. Okay, so genius just making come to talk about genetics later on. But anyway, so nurture is very, very important. Why do we have this forum here? We just want to have the young people in front of us study. And when you study, just study really the basic. Okay, you study broad and deep. You must know your basics very well. You don't just superficially know something and say, I can look it up in the internet. But when it really comes to solving a problem, you need experts. Okay, so therefore, nature and nurture work together. So when you say a superhuman being, I said, yes, there are some superhuman beings. Some can run very fast, right? Like your son, okay? And then some can be a math genius, and of course you have ways to discover them. But I don't think one should think about using genetics or using DNA to create a new superhuman person.
Yeah.
But education can discover or uncover a lot of talents.
Yeah, there's a lot of examples where nature affects, and nurture affects nature. So there's also a question for Professor Church. So how can the genome explain our behavior and non-physical phenotypes such as personality?
Even if we cannot explain the great diversity and nuances, there occasionally are cases where, at least in animals, we can explain a large effects. And so there are some examples of individual genes, sometimes combinations of 2 or 3 genes that affect very dramatically things like anxiety, ability to solve problems, mazes, and so forth. I would not call these super mice. They're just a little different, but they're very statistically significantly different. So there are things that we can do with single genes that even in the presence of thousands of other genes and environmental factors, one gene can have a big effect. And from an engineering standpoint, that's all that you need. So for example, for a variety of reasons, human growth hormone or somatotropin can be used to have a dramatic effect on height, even though height is affected by thousands of genes and environmental components. So I think the same will be true of behavioral, and we already see pharmaceutical substances that have huge effects on behavior, both positive and negative. So that will almost certainly be true of the gene products which those pharmaceuticals interact with. So.
See, genetics is a very interdisciplinary field now, and Professor Church, I guess, sort of reflect that in the variety of fields that you need to master or understand to develop the techniques you're developing. Now, talking to the audience, maybe future scientists, geneticists that are here, they understand, the importance of computer science and bioinformatics, the developing new tools and technology, big data science to develop personalized medicine, gene therapy. So what would you recommend in terms of training for those students in the audience who aspire to a career in genetics?
Well, first of all, I personally enjoy a large number of fields, but it's up to the individual. I think there's plenty of room for people to be very, very specialized. Not everybody has to be bilingual or more. But there are also there are plenty of opportunities. For example, in my field, we in addition to the subjects you already mentioned, computational and molecular biology, it's also handy to know optics because we're doing more and more super resolution. I think studying the genome through microscopy is extraordinarily important. Studying the epigenome at nanometer resolution is going to be critical and inexpensive. Economics obviously is important. In psychiatry, there's a variety of social components. So I think that there's opportunities, but you should follow in part where the new opportunities arise, and follow your heart in terms of what you like, but not just your heart. Look at the opportunities as well.
Completely agree. Dr. Tsui, any thoughts?
Well, no, I've been speaking to high school students a lot, okay? And then nowadays, high school students don't study science because science doesn't make money, okay? They thought they study business and other things that make money. And as a result, okay, they cannot do a lot after they study business because there's so many people. In fact, I would come back and say, if you want to be a scientist, the first thing you want to study is get your math. Okay, study your math well. Okay, it doesn't mean that you have to be a math expert, but please study your math. That's the number one thing. Math is the mother of all science. Even though math itself, I sometimes call it not really a true science, because scientists discover things and mathematicians just create things. They just invent things. Okay? So, but anyway, so have a good training in math. Okay, you can do biology, you can do chemistry, you can do physics, you can do neuroscience, of course, talk about computational science, and then computer engineering. Okay, so science is so very important. You study math. I mean, math is so important. That is science.
Thanks. In your view, as a personal perspective, what new surprises do you think genetics has waiting for us?
Well, I think George already touched upon that. Many common diseases. Now we are knowing the cause and the contributing factors, and some major and some not so major. A lot of reasons cause you say be forgetful, for example. Okay, aging is one thing, and it's not because our genes are changing, our functions are deteriorating. Okay, and then but say we know how to so keep a healthy life. Knowing our genes, you can avoid certain things that cause problems. Okay, so if you have a tendency to develop high blood pressure, then not to avoid the salt. If you have tendency to have diabetes, then you try to avoid sugar. And these information, okay, is already there in the genome, and then you can sequence and find out. But then I'm going to say these so-called common diseases, and it's not a simple one thing. Of course, there are certain diseases, even Alzheimer's, like a major gene. Okay, one major gene, you can have it, and by 30 years old, you develop, I think, Gonzalo, you remember that there's some history work, right? So one gene, it can cause you Alzheimer's. Okay, so therefore, I mean, a lot more information will be out there, and then how to use the information, I think, require quite a lot of understanding. So I think we're now only at the surface. Well, scientists usually, of course, we speak to scientists ourselves, but I now speak to a lot of general public. So I always come back and caution them about the genetic information. It's not just one thing leads to the other, and it's like a formula. You punch in and then you get a result. Okay, you really need some experts to help you. Okay, so the next thing perhaps is the information, the technology, helping people how to understand the genome, and then the level of understanding increases in the community. Okay, perhaps more people helping each other. So I think it's not going to be just one thing that will change the world, but then a lot of information, a lot of technology that will come together, and then we benefit from it. It's just like the COVID-19, the COVID-2 you're just talking about. We know the viral genome, and then the people say the vaccine is around the corner, right? So not so simple. The vaccine still a lot of development. And then when the vaccine is here, I hope the virus disappear by itself, just like a SARS virus some years ago, some 10 years ago. So the virus just simply disappeared. So that would be the best scenario. But then new viruses will come. So we just coming back and then deal with the nature. And then we have more and more information out there. And then therefore, the general understanding about genetics, general understanding about science is so very important for not just students going to universities or coming out to try to find a job, but it has to be everybody, especially the politicians who should understand more about science.
Yeah. Professor Church, any final remarks on perhaps some surprises that you think genetics might be waiting there for us?
Well, I think one surprise will be its impact on fields outside of biology and medicine. For example, DNA can store data. DNA can produce new materials, even materials that don't have carbon in them. Inorganic materials can be made by synthetic biology. So I think we're going to be surprised at how unlimited is the field. I think also we'll be surprised by how much we can do with just reading. It seems like reading is a very passive thing. It doesn't actually cure anything. But as we've kind of looked through this discussion, things like cystic fibrosis and COVID-19 can be fixed by reading, just by reading. And in fact, it might even be more effective than a vaccine or a small molecule drug. We just need to think very more cost effective. One of the biggest ethical issues is whether we have broad availability, whether you have accessibility to all people independent of socioeconomic status. And I think reading is already very affordable. It's on the order of $100 a genome, while it's millions of dollars for orphan drugs for a lifetime treatment. So we need to think about, we're going to be surprised by, and we need to think and encourage reading as a preventative, as a low-cost, equitably distributed technology. And then finally, I think we're going to get much better at building parts of humans, not just for transplants, but for enhanced transplants that are better than you could get from the healthy human donor. They could be better in terms of senescence resistance, pathogen resistance, cancer resistance, so forth. These things are going to be engineered into organs and very inexpensively. And that will allow us to test new therapies. It will allow us to test ideas in a multiplex fashion where you can test millions of different things at once. And we're already doing this, where we're testing millions of designs for viruses and cells simultaneously in one small experiment. So those are some of the sources of surprises, I think.
Yeah, of course. With that, I would like to thank you, Professor Church and Professor Tsui, for your perspectives, for your insight, and for addressing this audience tonight. Thank you very much.
Thank you. Thank you very much.
Pleasure.